in Berlin, and she's going to talk about how complexity arises from molecular interactions. And I think we'll hear something about microtubule dynamics, but I'm not sure. And so I'm curious to find out. So go ahead. I look forward to hearing about it and seeing it.
Thank you. Yeah, and thank you also for giving me the opportunity to talk about our work here. So as introduced, I will talk about complexity and how complexity arises from molecular interactions, in particular in the context of microtubule dynamics and the mitotic spindle. And one cannot understand complex collective phenomena by simply studying a simple component of the system, such as a bird or a water molecule. And this is because the interaction of many individuals gives rise to new emergent properties of the system. And this is true for birds and their collective behavior of flocking. This is true for the beautiful patterns that arise when snowflakes are built, dependent on the physical parameters of the system. And this is also true for microtubules that self-organize with many different proteins into a mitotic spindle. And such a mitotic spindle is shown here on the right. And a mitotic spindle is a molecular machine that assembles from microtubules with the aim to segregate our genetic material once every cell cycle. And this very process is going to happen several million times in each of your bodies, even during the time of this talk now. And the spindle's main building block are microtubules, and microtubules are polar polymers of alpha and beta tubulin. So tubulin will polymerize into microtubules, and they, with the help of microtubule-associated proteins and motors, will self-organize into a mitotic spindle. And this mitotic spindle, dependent on the cellular context, will have a different or a specific length and shape, and also orientation of the spindle within the cell. And while a cell type specific length can be robust and stable for several hours, the individual components of the spindle turn over on a second time scale. And this, I hope you can appreciate by looking at that movie where we labeled individual plus ends, growing plus ends of a microtubule by a protein called EB1. So really the microtubules turn over every about 20 seconds in such a reconstituted spindle. So really the question is, how can a structure have a very stable, steady state length when its individual components turn over on a second time scale? And length and shape are really critical and essential for spindle function. So these are emergent properties that are critical. And this is because the length of the spindle determines the distance over which chromosomes will be segregated, and the shape determines directionality. So the chromosomes will need to move into opposite directions onto the newly formed daughter cells. And we know of pathophysiological scenarios where spindles are either too short or multipolar, as here shown in the fluorescence movies. And these kind of scenarios, they can lead to chromosome missegregation and aneuploidy, which is a hallmark of cancers. When we think about spindle size and geometry, one can ask two very basic questions. And one question is about, or is the question about spindle scaling. So how does the length of the spindle change with cell size? And this question has been studied mainly in early developmental systems, in frog eggs or fish eggs. And here the advantage is that the eggs are pretty big and they have very rapid cell cycles without intermediate growth. And in these systems, it indeed has been shown that the length of the spindle scales with cytoplasmic volume of the cell in a given regime. The second question is within or outside of the cellular context is the question about size control. So what determines size per se? And such a system has been beautifully set up by Rebecca Hill's lab in Berkeley. There they have two different frog species, Xenopus laevis, which is a bigger frog, and Xenopus tropicalis, which is a smaller frog. And these frogs, they do not only differ in organismal size, but they also have different cell sizes, egg sizes, and hence also different spindle sizes. So the tropicalis spindle is only about 70% of the laevis spindle. And Rebecca's lab and a few other labs in the field, they've characterized molecular components that contribute to setting this overall difference in spindle length. And these components are all microtubule associated proteins. So proteins that influence microtubule dynamics or microtubule number. So we ask, what is the contribution of tubulin itself? Can tubulin itself contribute to setting the size of the spindle? And until very recently, this question could simply not be answered because we were lacking Xenopus tubulins. Because classically, in the last 50 years, the microtubule and spindle field has mainly relied on bovine and porcine brain tubulin, because tubulin is quite a tricky protein that cannot just be overexpressed, or you cannot just put a his tag on tubulin and express it in E. coli. So classically, we had to go to the slaughterhouse and get pig or bovine brains, because brains and neurons are very nice source for tubulin with all the long and stable axons. However, this heterologous tubulin, of course, has a few disadvantages. And what annoyed me mostly was that, we're working on cell division, we're working on spindle assembly, but we only were working with tubulins that had been purified from non-dividing cells, which are neurons. So during my time in Dresden, we we established a protocol which allows us to purify tubulin from every source. And by now we have purified tubulin from many different species, tissues, and even parasites. And here, this is an example of where we purified frog tubulin, and importantly, all these tubulins are functionally assembly competent, so they build microtubules in vitro, which are dynamic. So now that we purified tubulins from the different frog species, we could go back and answer the question, can tubulin, the main building block of the spindle, can it potentially also contribute to setting the size of the spindle? So what we did is that we in vitro reconstituted microtubule dynamics. So in all these assays, they only have the purified protein tubulin. So there are no additional other proteins. And this is going to be important later. So here you see a so-called turf assay where you stick individual stabilized microtubule seeds to a cover slip, and then you can flush in this case, green labeled tubulin, and observe how the microtubules show a behavior which is called dynamic instability. So what this means is that they grow and they shrink, and they stochastically switch between these parameters. And when they switch from growth to shrinkage, this is called a catastrophe. And when they switch from shrinkage to growth, that's called a rescue. And from these kind of turf assays, one can measure, quantitatively measure these parameters of dynamic instability. And here you see just for now individual microtubules. So this is a chymograph, which shows a single microtubule over time, so length over time. And what you might already appreciate from the single microtubule is that, so Xenopus laevis is our big frog and Xenopus tropicalis is our small frog. So what you might appreciate from this single chymograph is that the dynamics that we see are quite different. So the Xenopus, the big frog microtubule, they grow faster and they catastrophe not very frequently when compared to the smaller frog microtubules. And these parameters we can quantify, and what we see is that Xenopus laevis microtubules grow almost twice as fast as Xenopus tropicalis, and also they almost never catastrophe. And in the very, very few cases we saw them catastrophe, they rescue with a way higher rate than, again, tropicalis. And then we also always show bovine tubulin as a comparison, because this is what the field is used to looking at. So the Xenopus laevis, the big frog microtubules, grow faster and catastrophe less frequently. But how can these microtubule dynamics influence spindle size? And back from my postdoc times, we developed a mass balance model, and here only for now 2 parameters are important. One is the growth velocity that we've measured of microtubules and the lifetime. And what you can see here is that the length of the spindle will depend on lifetime and growth velocity. So if we increase these parameters, we will expect more microtubule mass and therefore a longer spindle. So what we did next, first in our turf assays, that at the same conditions, so this means same concentration, same temperature, same buffer, we just quantified how much of the tubulin was actually in polymer mass. And as you would expect from the microtubule dynamic parameters, at the same concentration, more tubulin is in the polymer mass when you compare it to Xenopus tropicalis tubulin. We can mix these tubulins and then they show an intermediate behavior. So what we did here is that we mixed 50% Xenopus laevis and Xenopus tropicalis, and nicely the behavior is titrable. So then the microtubules show an intermediate growth rate and they also show an intermediate polymer mass. So if I now, talk about tubulin, it is a protein. Where can these differences actually come from, right? But tubulin is not just a single protein. It is actually a heterodimer of alpha and beta tubulin, and frogs have 6 different alpha and 7 different beta isoforms. And in addition, the tubulins are heavily can be heavily post-translationally modified. So we know phosphorylation, methylation, acetylation, polychlometylation, and so on. So what we next want to check whether the tubulins that we purified and that show these differences in dynamics, whether they are molecularly any different. So we first looked at the isoform composition by whole protein mass spectrometry. And as you can see here, so again, big frog tubulins are in blue and smaller frog tubulins in red, is that the isoform composition of the 2 tubulin populations is qualitatively very similar. And then we checked for post-translation modifications and by Western blot, and as you see here, the bovine brain tubulin is heavily post-translation modified. So this is something that has been known. And compared to this, frog tubulins are almost not post-translationally modified. And we only find a single phosphorylation that is present in Xenopus laevis microtubules, but not in Xenopus tropicalis microtubules. So that is the only difference when we compare the 2 tubulin populations that we purified. And of course, this now is something that we're really trying to figure out, whether this single phosphorylation can make all the differences in dynamics. So, but now, what about spindle size and spindle geometry? Can the dynamic parameters that we measured, can they really change the length of the spindle? So we first used cytosim to model spindle assembly in silico, because here the nice thing is that you can individually modulate all individual parameters of microtubule growth independent of each other. And just by tweaking microtubule growth velocity, we can establish the difference between the sizes of the spindles, and this is something that has already been shown again by Rebecca Heel's lab. And also, if we swap microtubule growth velocities, we can re-establish the size in the individual spindle assembly reactions independent of the cellular environment. If we now go back to in vitro reconstitution, the best way here to test whether the tubulin can contribute to difference in size is that we assemble spindles in vitro and then titrate in increasing amounts of tubulin and measure the length of the spindle. So here we pre-assembled big frog spindles, Xenopus laevis spindles, and then we titrated in different amounts of bovine brain tubulin. And here we could not see an increase in overall spindle length or size. And this has been something that Rebecca Heel already has reported in a paper they've published earlier, where they hypothesized or suggested that tubulin would be a limiting component of spindle size, but also they could not, by increasing the concentration, show that this would increase the length of the spindle. However, if we now take our Xenopus tubulins and increase the concentration of Xenopus-specific tubulins, what we see is that the length and the mass of the spindle increases. So again, this is the Xenopus egg extract, so proteins, microtubule-associated proteins. The cellular context is from the big frog, and we titrate in small frog tubulin and big frog tubulin, and we see that the length of the spindle increases, and it increases slightly more with the big frog tubulin. So this is even more obvious if we now start off with the smaller frog tubulin, right? So now we take the cellular context of a Xenopus tropicalis extract, assemble a spindle, and then we titrate in small frog tubulin and big frog tubulin. And as you can appreciate, when we now use the more dynamic or the faster growing Xenopus laevis microtubules, then we get quite a dramatic increase in microtubule mass and also in spindle length. So this all, summarizing the first part of my talk, this all led us to hypothesize that really tubulin itself has some intrinsic properties that allow for different microtubule dynamics. So in this particular case, it would be the growth velocity and the frequency at which microtubules catastrophe. And these differences in microtubule dynamics, they will contribute to the polymer mass. So there's just going to be simply more microtubules, which will then lead to a bigger spindle in the bigger frog. So of course, the other proteins that have been reported, like TPX2 and catalin, they will contribute. So, but tubulin itself should now be considered as one of the players in that system. So, what I haven't talked about so far is temperature. And we know that microtubule dynamics are very temperature dependent. And the frogs in the lab do live at different ambient temperatures. And now we even have a third species. We now also have Xenopus borealis that lives somewhere within in between the temperature range of Xenopus laevis and Xenopus tropicalis. So we have 3 frogs that live at different ambient temperatures, and as frogs cannot regulate their body temperature, this means that the microtubule dynamics and also cell division need to happen at the ambient temperature at which these frogs live. And laevis lives around at around 18 degrees, borealis around 23, and tropicalis around 28 degrees. So they spend almost 10 degrees temperature range. Now we've purified tubulin also from borealis, and now we can go back into our in vitro system, the turf system, and measure the dynamics. And Anatol Fritsch from Dresden has built a really nice sapphire stage with which we can robustly control temperature over time, and also allows us to change temperature through a single turf assay. So this means that we can look at microtubules. We, for example, can start out measuring microtubule growth at 25 degrees. We decrease the the temperature, and in this example, we would not see any growth at 22. We increase the temperature to 30 degrees, see an increase in microtubule growth velocity. And importantly, we use to end the experiment at the same temperature we started to compare the growth velocity to make sure that we did not exhaust the tubulin, because such an experiment takes about 1.5 hours. So we can do this kind of experiment at very different temperatures, at very different concentrations for all our species. And then we can take the mean growth and plot this against the tubulin concentration. And here we've done that, for example, for Xenopus borealis. And then from these growth versus concentration graphs, we can determine the rate of microtubule growth at each temperature. And then again, we do this for all 3 species. And then with the rate constant, we can use Arrhenius to look at the activation energy of microtubule growth for the 3 species. And what we see, so this data set is not yet complete, so we're still planning on repeating a few of these experiments, and all is not published yet, but there's a clear tendency that the biggest frog that lives at the lowest temperature has the lowest activation energy for microtubule growth. And this is really, if you think about a microtubule and the microtubule plus and the soluble tubulin that needs to come in and incorporate into the microtubule plus, and the activation energy of this process is lower for the species that lives at the lower temperature. So these are now the data for the growth velocity. We also have them for the depolymerization velocity, for the catastrophe and the rescue frequency, and also for the critical concentration. And we're using these parameters on the one hand to build a theory that might explain how we get to these different dynamics with these different activation energies. And this we're doing together with Gary Bruhart, but also, and this already takes me to the outlook, we would like to understand how we can explain this molecularly. So do we see differences really in the sequence of the individual tubulins? Can we see the difference in the course of evolution? So when did certain potential substitutions really appear? Is there a thermal optimum for our species? And then related to that, are there thermal limits? So is there a break-off point at a certain temperature where we do not see functional microtubules and therefore potentially cell division? This also for is limited that Xenopus laevis and borealis are not sequenced or not really entirely sequenced. So this is something that is limiting us a bit at the moment. Yeah, so now I'm ending on a negative note, but that's, we're still trying to figure this out, and we're optimistic that we'll find also molecular explanation for the differences in the activation energy. So with this, I would like to thank our collaborators, without whom we would not be able to do what we do. Thanks to all of them, and also, of course, to the people who did the work. So Abin and Will are the two students who worked on the first part of the story on the size difference in the reconstituted spindles. And Ella is the student who is working on the temperature-dependent differences of microtubule dynamics in the 3 frog species. So with that, I would like to thank you, and I'm happy to take questions.
Thanks a lot. That was a beautiful and very clear talk, I think. Yes, nice. Questions from the audience. Let me see. Ayan, go ahead.
Yeah, so Simone, that was a lovely talk. So I was just wondering, so you have mentioned from the first part of the talk that the dynamics, the microtubule dynamics is really species dependent. And given the fact that the tubulin accumulation at steady state are different for those 2 species, I was just wondering if you have taken into that fact into account. For example, if you had a normalized statistical metric for the dynamics, maybe you can get a universal feature popping out from the dynamics. Maybe they are not different. If you take into account the size effect of the accumulation of tubulin. So have you done that? I was just wondering.
So your question basically is whether, you know, whether the difference in dynamic we see is physiological relevance, right? So the effect size of the differences we see. And of course, this is always a question, right? If we just increase our measurements, we'll always get a statistically significant difference, but we also do measure effect sizes. So we've done that, right? And I would argue that having twice, in particular growth velocity, right? And this is also then reflected in the simulations. If you double growth velocity at which microtubules grow, you know, that really has an effect. And I think the strongest argument is our reconstitution assays, where we add in these different tubulins and we see differences in increase in the final spindle. So I think, you know, this is an experimental answer to your question, I guess, but I think that is the strongest argument that a difference in tubulin biochemistry, which we definitely have, you know, within these 3, contributes and is therefore physiological significance.
Thank you.
Christina, you had a question?
Yes, yeah. So Simone, beautiful talk. I really enjoyed it a lot. I've been in touch a lot with microtubule people in the past, and I think post-translational modifications have of course been debated already heavily for quite some time. So it's really cool to see the data. And I was just wondering, now you have access to tubulin that is basically not modified. Is there a way to in vitro add modifications so that you could like step-by-step reconstitute what these modifications do?
Yeah. I mean, yes, and that is something that we have one project where we work on detyrosination, for example, because this is now tubulin that is not detyrosinated naturally, right? So it's a nice substrate for for the enzyme that detyrosinate. So yes, we can do that. However, being critical about our system is that it always is a combination of different isoforms. And we do not yet fully know for like the complete systematic qualitative analysis. Ideally, what we would also love to know is the percentage of isoforms, right? So if you look at the different isoforms, you know, it would be great to know, you know, we have, I don't know, 50% of A1 and 20% of A3 and so on. And this so far is not so easy. We're trying to do that. But then if we really have, you know, complete quantification of isoforms and PTMs, then that is even more feasible. Yes. Yeah.
Cool.
More questions?
And oh, sorry.
Sorry, go ahead.
And therefore we have now very simple systems, like particularly parasite microtubule populations that are very simple, where you only have one or like 3 or 2 isoforms, right? And that, of course, these systems are interesting because there it's easier to then really study the contribution of individual isoforms, for example. So it turns out the simpler the system, the easier to ask such questions. And yeah, Xenopus is rather complex. Yeah.
I don't immediately see other hands, so I might ask something myself. So you talked about the influence of temperature on the growth of microtubules, and you've shown like that there are different activation energies in the different species, at least for the parameter that you've shown. Do you have some ideas of why that may be, or can you speculate on that?
Yeah, so far, no. There is one report by Gary Bruhard's lab who looked at C. elegans tubulin, which also has a lower activation energy as compared to the bovine tubulin, which we're all used to looking at. And what he reports a structure of the C. elegans tubulin, and his group reports is that it has a higher free energy, right?
Okay.
So this is something that we're also interested in. However, looking at like, you know, imposing structure is difficult, particularly for Xenopus laevis, because of lacking sequence, and also Borealis is not sequenced. And what we're might also, you know, be now looking at is, you know, just doing cryo-EM of the microtubules that we can assemble at different temperatures and maybe get some structure information from that.
Mm-hmm. Okay. I still don't see any other hands, and we still have 5 minutes, so I might just chat a bit more. Sure. So you're mentioning one parameter here. You also said that you measured some others. Do they follow the same trends, or do you see different behavior there?
So what we see is that the other parameter that's mainly affected is the rescue frequency, and one parameter that is unrelated to the classic dynamics, which is the critical concentration, which is the concentration at which the microtubules will grow. And also for the critical concentration, we see quite a difference in that it's way lower for Xenopus laevis. But the overall picture is quite consistent. So because we see a lower critical concentration for Xenopus laevis, so they will start nucleating and growing at a lower concentration in the colder environment, and they will grow faster at comparable temperatures. And if they happen to catastrophe, like, you know, fall apart, they're more likely to rescue. So and these are all parameters that will make microtubules more stable at colder temperatures. So yeah, although, as I said, you know, it's not, we need to repeat a few experiments and a few temperatures and you know, put in a few more concentrations to make this really round and quantitative. But yeah, so the picture that we're seeing is consistent.
very nice. So if somebody else has any questions, just yeah, give me a nudge or something. I'm trying to keep
breakout room.
What, sorry?
If not, I'll be in the breakout.
Yes. Okay, good. Well, we still have 3 minutes. So maybe I want to ask maybe about your first part, where you sort of analyzed spindle size and how that depended on the tubulins. And so at some point you were also talking about spindle geometry, but then I saw mainly something actually about more size. So I was wondering, did you really look at the mass and the size, but also at, did the geometry itself change as well?
Yeah, so this is something that, or is it maybe a slide, that we're interested in, and that we also have projects where we look at the shape and also at the orientation. So these are basically 3 properties of the spindle that interest us. And so the idea we have when we talk about spindle length and shape is that the length of the spindle basically is determined by its mass. So simply, the more microtubules you have, you will make a bigger spindle, but the shape will not change, and that also has been shown. So that's why our hypothesis or our argument is that the length of the spindle is mainly determined by mass balance. So whatever is going to influence microtubule dynamics will have mainly an influence on the length of the spindle. The shape is determined by a force balance. And that also has been reported by others. And we also know, of course, of phenotypes where you can massively change the shape of the spindle. And then there is one parameter that will most likely change both, and that is nucleation, right? So if you change microtubule nucleation, this, based on theory and on experiments we've done, supposedly is going to change length and shape. Because of course, increase in microtubules and the nucleation profile can contribute to the shape of the spindle. So this is how you can tweak these a bit apart.
Right.
So here, if you mainly have an influence of microtubule growth, you would not necessarily expect a change in shape so much.
Okay. All right. Again, thanks a lot for a very beautiful talk.